transforming principle is one of the most important scientific discoveries in all of mankind. 3 scientific researchers contributed to this discovery: Oswald Avery, Colin MacLeod, and Macklin McCarty. These 3 scientists are responsible for the discovery of DNA. However, they did not come to their discovery without some help from a fellow scientist's research that took place 20 years prior to their discovery. We will start our journey to finding DNA with scientist Frederick Griffith. Griffith was working with the bacteria that is responsible for pneumonia. He used 2 different strands, the R strand and the S strand. The R strand was non-infectious, rough, and lacked an important enzyme found in the S strand. The S strand contained a polysaccharide coated and had an infectious agent. Griffith heated the S strand up and injected small particles of it into the non-infectious. Only the R strand was injected into a mouse, it would live. But when the R strand with some S particles was injected into a mouse, it would die. This caused Griffith to be unable to comprehend what was causing the transfer of deadly S particles into the non-infectious R strand. This is where Avery, MacLeod, and McCarty come into play. They had begun to study and review Griffith's research and were interested in finding the material responsible for the transfer of infectious genetic material. Avery began the discovery by a process of placing the bacterial strand into a centrifuge, heating it up, and thus killing the bacteria and extracting the different components. The next step was to place an enzyme inside the test tubes that would destroy the RNA, DNA, and proteins. The 3 scientists were stunned when they discovered the transforming principle could not be broken down. They first discovered the transforming principle was not a carbohydrate when they placed it into alcohol and were unable to break it down. They then placed the transforming principle into the enzyme protease, thinking it was a protein and trying to break it down. Again, they were unsuccessful. Their last attempt was to break it down in the enzyme lipase, hoping that it was a fat. Their last attempt was unsuccessful as well. The 3 scientists continued to study the transforming principle and were able to discover that it was rich in nucleic acids, had a high molecular weight, and lacked a ribonucleus. They decided to test it in DNA one last time, and this time they were successful in breaking it down. Therefore, they were able to conclude that the transforming principle was in fact DNA.